Okay, everybody. Anybody have any questions before we start?
Will we be having a quiz before the next test?
Will we be having a quiz before the next test? Yes, you'll have one the day before the class, before the exam. So we'll go, yes. The quizzes are always loaded toward the exam. Any other questions? I didn't put my question up. I should have done that. Okay. Okay, let's pick up here again, and I want to add some complexity to this. We finished here last time with the fact that there are these introns that are removed, and that the RNA, even after this step is processed, it has a string of adenines added onto the end, so he call this a poly-A tail. And then we have a fairly complex cap added to the other end. And then these introns are removed, and then the piece of DNA is put back together, and then it's moved out of the nucleus where translation can occur, and this is ribosomes, of course, attaching to the RNA. And today we're going to start talking about, or talk about the fact that there's other genes that are what we call regulators, regulatory genes. And so we have genes that control whether or not RNA can be made. And these are called regulatory genes. And then these genes that make a product, that actually make RNA, that eventually is converted into proteins, are what we call structural genes. So what you're going to see today, we're going to talk about regulatory, and these are genes that control whether or not the RNA is actually transcribed. And then if we make a gene product, if we make a protein, these are called structural. Structural genes, okay? Now, here's just another cartoon showing you what we went over this last time. And then this is the 5 separate levels of control that determine whether or not the DNA template is actually expressed as a protein, okay? So we have the genome, of course. Then we have transcription, which requires RNA polymerase II and a lot of other enzymes. Then we have processing and translocation. Processing is when the 5' cap is added, and that string of adenines, of A's, is added to the other end of the RNA molecule. Then it has to move out, so the nuclear pores have to be open so that that piece of RNA can move out. And then it's translated, and then we have another one here at the bottom called post-translational. And look at these. We've talked about all of these. It has to be folded correctly. It may be cleaved. One of the most common things that happens in post-translational is we have an ATP and we add a phosphate to the protein. So And then the take-home message is that this is an incredibly complex process. And you've heard of understanding something like peeling away the skins of an onion. So we learn something and that first layer goes. We learn something else and the second layer goes. When it comes to this, every time we learn something, we add a layer to the onion because it becomes more complex. In this, you have the processing and RNA polymerase II. Okay. As though RNA polymerase II, the polymerase, the enzyme that makes RNA, is like the standalone enzyme that does everything. No. There are another 20 or 40 enzymes / proteins that are necessary for this to occur. So we understand more about a single gene, say RNA polymerase II, but the onion just builds more. The more we know, the less we know. Well, that's not right, but the more we know about RNA polymerase II, the less we know about the new proteins that we did not know existed until those experiments were done. Incredibly complex. That's incredibly complex. Questions? Okay, now, this is called the lac operon. And this was the first, and why I use it, this is the first gene in which the regulation was figured out. And you may have heard of Jacob and Monod, who were two French researchers that did this. Anybody ever heard of Barbara McClintock? No one heard of Barbara McClintock? Barbara McClintock won the Nobel Prize in the 90s, maybe, for work she did in the 40s, where she showed that pieces of DNA would move around in corn. That a piece of DNA would actually come out of the gene, come out of the chromosome and move around. And nobody believed her. The reason why I mention her is because she was in their lab when this was elucidated. And I think it was Monod that was an anarchist and was in prison most of the time. So there's this notion that maybe Barbara had something to do with it, figuring it out, how it worked. And so we're going to spend some time on this. I like to cover this because this is not exactly what happens in plants, but it's a really, really good, simple gene regulation system. Okay? And we have a lot of players in the system here. And we also have changes in the three-dimensional structure of things. And we're going to start from the left, which is a regulatory gene, which can be somewhere, anywhere. And this regulatory gene produces, from transcription, a repressor protein. And in this cartoon, this repressor protein has a certain three-dimensional shape that's flattened out to be this shape. And this repressor protein binds in a site called the operator. Okay, the operator. Okay, now, how is RNA made? If we go back here, we have the RNA polymerase that's here in the promoter. And here in this cartoon, we have an RNA polymerase that can only bind in this other regulatory gene called the promoter. So RNA polymerase binds, and then it starts moving down the RNA, and it runs into this roadblock. Okay? Can't make any RNA. Just runs into that protein and it falls off. Does that make sense to you? The RNA polymerase actually moves down the piece of DNA, and as it literally moves down the DNA, it's producing the RNA behind it. Okay? Now, let's talk about what this operon does. This is a bacterial operon called the lactose operon. And what the lactose operon does is you have your bacteria growing and you add lactose to the bacteria, and it's able then to break down lactose, milk sugar. Okay? So the genes are only expressed, and that was what they knew before they did these experiments, is you only find these three structural genes, Z, Y, and A. You only find those gene products produced, those proteins produced, if lactose is present. Now, that makes sense, right? The cell is not going to waste energy. So if there's no lactose present, none of these genes are produced. You don't need them. Why would you have a gene to break down lactose if there's no lactose around? Okay? And so that's what these folks knew when they started doing these experiments. So under conditions where there's no lactose, you have this repressor protein produced, and this repressor protein binds to a site called the operator. And then the RNA polymerase binds, and it runs into a roadblock, and it falls off. Okay? Questions about that? So you have regulatory genes and you have structural genes. And these three structural genes, so if we go back, these are structural genes. These genes become enzymes. Genes Z, Y, and A are needed to actually break down lactose. Okay? Now, what I've done here is put the first one we just talked about there. And now at the bottom in B, we have a system where lactose is present. So now you've taken the cells and you've shot in some lactose. And we know that bacteria can live on lactose alone. And that if you add lactose, and this is a very, very important thing, you induce the production of the genes needed to break down lactose. Okay? Understand that? You inducible. This is an inducible gene system. Questions? So how does it work? Just like we've talked about previously, the inducer is lactose. So the inducer, write that down, inducer is lactose. And guess what? Lactose binds to the repressor protein, and when it binds to the repressor protein, it warps the three-dimensional structure of the repressor protein. And let's move up to this top panel. You have the operator. binding the repressor protein. Now you have a repressor protein that's warped, which means what? Given what you know about how proteins work and how things can, it won't fit. It will not fit, okay? So now the RNA polymerase at the promoter, and of course you need to know the promoter, the RNA polymerase binds to the promoter and now moves through the operator and makes RNA. And the RNA that is made is the 3 genes that the bacterial cell needs to metabolize lactose. Okay, isn't that cool? This is the first one ever identified, the first gene regulation system that was ever identified. So at any given time, you've got the RNA polymerase binding to the gene called the promoter of transcription. And in the top panel, it hits the repressor and falls off. It can't go, can't move anymore. Then in the bottom panel, we've added lactose. Now, and when we add lactose, the lactose binds to the repressor in such a way as to warp it to where the repressor can no longer fit on the DNA. Okay? So, the bottom line is we have these bacterial cells growing in sucrose or glucose. So they don't need the enzymes to break lactose down. That would be a waste of energy. You've all heard of gene regulation. Okay, in this simple bacterial system, it is gene regulation. So if the cells don't have lactose, it's a waste of energy to produce those enzymes needed to break down lactose. So in the top panel, there's no lactose present. So this repressor protein binds and those genes are not produced. Takes a lot of energy to make RNA. It doesn't make any sense to make the RNA if it's not needed. So this is a way that we shut down RNA production. Then if lactose is entered into the system, if we add lactose, the lactose then binds to the repressor, and then this genome can be transcribed. Okay? Questions? So what happens if you have a mutation so that the operator is gone? The operator's been removed. It'll always make the gene products. So think of it literally as the RNA polymerase binding to the DNA and starting to move down, and it runs into a roadblock. If we take that roadblock out, then that RNA is going to just go ahead and continuously produce those genes. So even without lactose, we produce the ZYNA gene. Okay? Now, we're going to finish up today with a topic that's going to cover everything that we've covered up to today, from the first few lectures all the way through today. And it's about bourbon. You know, we've got some bourbon places in Texas that that actually make. I always thought bourbon had to be made in Kentucky, but apparently not. Anybody watch that really bad show called Moonshiners? You know, where they're always trying to build their still and make the bourbon. There are some fairly specific requirements for something to be bourbon. It has to be made with over 50% corn. And then you get into the straight bourbon and sour mash. And if you've ever heard of sour mash bourbon, the thing about a sour mash bourbon is you take part of the mash from the first run and you use it during the second, and then so you keep transferring what we're going to talk about today, which is the yeast. And what you're doing is pretty simple. You're taking corn and you're soaking corn to release the starch, which is what we're going to talk about. And usually you add a very specific yeast. If you're making moonshine, which I don't think these guys are really making moonshine on Moonshiners, you add a yeast that you get. Mainly you add the yeast so you can get the same kind of product every time, as opposed to letting whatever the wild yeast is that comes in, you know. It's a little bit different in that because mostly I know about wine, is that if you see a place that's storing bourbon, the barrels are not inside a temperature-controlled environment. The barrels are put out in the weather. Okay, so I mean they're not rained on, but part of the bourbon aging is the differences in temperature. So they're pretty extreme in Texas. Anybody ever been to Jacques Daniel's? Yeah. So goes in the barrel, stays there for at least 2 years. And I think I've got some more pictures to show you about that. But here's a place over in Garrison Brothers. This is over near Fredericksburg, and this is just a vat of the corn that's fermenting. Now let's talk about where corn comes into it. And what you see here is a cartoon of a corn seed or a grain seed. It doesn't really matter what kind it is. We'll talk about corn. And we're going to talk about the production of an enzyme that's needed to break down the corn starch. And as an overview, let me show you the players here, is that if you take a corn kernel and look at it, what you can't see is that there's a single cell layer around the corn seed that's called the aleuron. And that's a single cell layer that's alive. In other words, when you put water into the corn, that layer has a nucleus and it produces proteins. And if you look inside the seed, and if you know, and everybody knows what the endosperm is, the endosperm is just a bag of starch and protein. It's dead tissue. So if you take a corn seed and cut it through the center, then the little white thing at the bottom is the embryo proper, is what we call it, the embryo proper. And it's the part that's blown up here. Okay? And then you have this bag of starch and proteins. And then you have this aleuron layer. Okay? Now, what happens is when you add water to the corn seed, the embryo starts producing a hormone called gibberellic acid, which you've probably heard about, okay? And then the gibberellic acid diffuses into this aleuron layer, which is this blue area, and the gibberellic acid produces enzymes that break down starch. Now, starch is what? Anybody remember what starch is? Nobody? Bunch of glucoses and an alpha-1,4. So why would the seed want to break down the starch? What are those glucose molecules used for? Huh? ATP production where? In the embryo. Okay? So the green thing, the embryo there, is going to need energy to grow, right? So what this seed does and what most other seeds do is they have that lot of stored starch or protein. So they break that starch down to glucose, which moves into the embryo, and ATP production happens. Metabolism happens, and the embryo can grow. Okay? So if I take a dry seed, nothing's going on. If I take a seed, and are you familiar when I'm talking about when you take and cut a corn kernel in two, you get the, and I don't know if I have a picture, you get the little bit that's the embryo at the bottom. 99% of the rest of the space is just bags of starch, dead tissue, and then surrounding the whole seed is that one layer of living tissue, the aleuron layer. Okay? So when you add water to the seed, it starts producing gibberellic acid, which diffuses to this one layer, which produces alpha amylase, which breaks down starch, and then the products then are moved into the embryo so the embryo can grow until it penetrates the ground and it can start turning green and producing its own carbohydrates. Okay? Now, here's what we see. We can take corn seeds and we can remove the embryo. In fact, a very easy way of doing that is to give rats the corn seeds. And the rats will eat the embryo, but not the rest of the seed, preferentially. So we can remove the embryo, or we can go in with a scalpel and we can dig most of the embryo tissue out. And if we do that, then we can then take that leftover part of the seed that has the aleuron layer, and we can add GA. And if we add gibberellic acid, which is a hormone, then you get alpha amylase. Okay? One kind of starch that you know is amylase. That breaks down the starch. Amylase breaks down the starch, produces glucose units. Okay? And you may have heard the amylopectin or amylose, which is different names for different kinds of starch. So if we artificially remove the embryo and we add GA, we get this. We get the production of alpha amylase, and we also get alpha amylase RNA. So if I take that seed that has the embryo removed and I put it in water, put it in a petri dish, nothing happens. The embryo's gone. Then if I take that leftover part of the seed and I put it in a petri dish and I add gibberellic acid, then I get not only the production of the protein, but I get production of the RNA too. Okay? Gene regulation. Gibberellic acid induces the production of alpha amylase. Okay? Let's go back one. Gibberellic acid diffuses from the seed into the alburon layer, and in the alburon we have a change in gene expression, which produces, among other things, alpha amylase, which breaks starch down to small molecules, glucose, or maybe 2 glucose units, which can then move into the embryo, and we know that glucose is used to produce energy. Glucose is the central part of metabolism, of glycolysis, TCA, ATP production, so the embryo is able to grow. Okay? Does that make sense to everybody? Okay. So we again use this GA to induce all these systems. Now, what's it got to do with bourbon? Okay. Anybody ever made whiskey? Anybody ever tried to? Oh, okay. So you have a bunch of corn, and corn is just starch. And what you need is to produce glucose, so the glucose can be used by whatever yeast you use to make alcohol. So you germinate the corn, and one of my students, I have a PhD student that runs a brewery in Houston. He's probably going to give you a lecture on brewing. What you need to do is convert that starch into glucose. So, and mostly what you'll see people do is they'll take those bags of corn and soak them in water, and the water, or heated water, hot water, and the water then breaks the starch down to glucose, which is what the yeast needs to make alcohol. So you have a dry seed here, and you add water to it, and you end up with then glucose, which the yeast can use to convert into alcohol and other things, CO2 and alcohol. You'll have several lectures on this conversion. You know, I teach winemaking, so this making bourbon is not really making wine. It's the same general principle, though. So you produce alcohol. And if you watch that series of moonshiners, they always have these incredibly bad stills that they've made, because you've got to distill it. Okay, so you've got 12 or 14% alcohol, but to make a bourbon or to make something like a bourbon, you concentrate the alcohol by distilling it. Okay, right? Everybody understand that? And by the way, that's something you can't do. You can make wine at home. You cannot make distilled spirits. And so if you do it in the class, you have, you know, you've all seen these. These are, you heat this up, and the alcohol comes out, goes down this condensation tube, and you collect the alcohol. Okay? When I was in high school, we tried to do this, and we didn't have these really nice tight connections, and we had this huge container filled with rotting peaches, and it was terrible. It was nasty stuff. And we decided, this is a project we were doing, so we were going to distill it. And this is a condensation flask. Well, the ones that we had, you connected up with rubber tubing from the flask that you heated up. And we didn't know what we were doing. And at that time, burettes didn't have the little stopcocks that you turn and close. What they had was a piece of rubber tubing with a bead in it. And when you would squeeze the bead, then a drop would come out. I didn't know what I was doing, so I pulled the bottom off a burette, and it was rubber tubing, and I hooked up the distillation flask, or the distillation column, this has cold water running through it, to the boiling flask with a piece of tubing that had that bead in there, which meant nothing could go through it. It was a closed system. So what happens if you heat up a closed system? Well, what happened was that, and we didn't have, we had a thermometer on the side, is that this was sealed up and it went pop. So the rubber stopper went pop. So what do you do? Well, you just ram it down in there as hard as you can so that it won't pop again. And so we're standing in the back of the room watching it and watching the temperature come up. All of a sudden, boom! We had this large container filled with rotting fruit. It was not fermented. It was rotting. And all of a sudden, it just goes sprays up into, and hits everyone in the back of the room. Now, who sits in the back of the room in high school? The football players. And so they were covered with this nasty crap. It stunk like hell. And so not only do we have to run away, we had to run away for a few days. So, but you can do this, and you'll produce some kind of an alcohol. It's a lot of problems to go through. Wine's a lot easier. And I love this picture. This picture is actually from a still in Africa. And it's basically the same thing. You've got the mash, and you're heating this up, and then, of course, the alcohol boils below water. So that's the beautiful thing you can do, is you only have to heat it up to about 80 degrees C, and the alcohol comes out, and the water stays behind. That's what you're doing when you're distilling. And this is pretty cool, because they actually this is called jet fuel, and they literally add jet fuel to the brew. And these are several steps. If you watch moonshiners on TV, they're not this elaborate. But you're distilling it, so the alcohol is going through these containers, and then if you're making vodka, you may redistill it several times. So you're distilling it to get the alcohol, and then you redistill it to remove some more contaminants. And then you end up with something that's drinkable. And this is over in Fredericksburg. This is Garrison's Brothers. They make bourbon. And this is this is several years ago. This is back when they've got much larger stills now. This is what a real still looks like. And as you heat up the mash in the bottom, you have these different layers or different levels where you can pull things off. Like if you know anything about how they crack petroleum, So, and in this case, they were actually collecting the bourbon in a pitcher, the alcohol. They were collecting the alcohol in a pitcher. So they, it wasn't like they were producing 100 gallons a minute. They're producing a lot more now, but those different levels in the stills will let you remove different products. And if you see at the top here, you have this line, this copper tubing coming down, and so whatever gets to the top, which you hope is mostly alcohol, then comes down, and these guys are literally collecting it in a pitcher. And They've got several stills now. And if you ever go over to Fredericksburg, you can tour the distillery. And here again is the mash. So now what you've got is the corn and water. So what you're seeing there is the gibberellic acid breaking down the starch to glucose. You've got to do that before you do anything. Okay? Does that make sense? And then here's their newer still. If you can see, it's much, much bigger now. And here's a very simple still. You put the mash in here and boil it, and then you collect everything that it comes out of the boil and comes down this tube, and so you have your container here, you know. So you're getting a pretty nasty mix of different kinds of products. But the basics are still there. You have your corn that you have had to treat so that the alpha amylase, the other gene products that are required to break down the starch are produced. And then you get your, I guess this is a home style still, I don't know. Okay? Then you age the alcohol for at least 2 years. And here's a cross section. Here's the barrels in one of their barrel rooms. And the inside of the barrel, most people don't know this, but when you buy a barrel, the barrel is toasted inside. So a barrel is put over an open flame, and the interior of the barrel is burned. It's called toasting, and you do this for wine too. With a bourbon barrel, you really toast it. That's where the brown from the bourbon comes from. Alcohol is clear, okay? So this toast then gives you very specific flavor components to the bourbon. And then you put the alcohol in here, and I think for bourbon it's still 2 years. And that's not a great picture on the right. You can see it's black inside. That's because it's been fired. It's, you know, you literally put it over a fire, moved it around. And then, because you're producing alcohol, what you get from a still is some flavor, but mostly alcohol. So you get a little flavor, so people use bourbon, they use other corns, or they use other kind of materials in there to give you a little bit of, because you're not only getting just 100% alcohol over, you're getting some flavor coming over. And then when you age it, you're pulling out the oak and you're pulling out the char into the bourbon. So what goes into this barrel is a clear liquid. They can be made out of different woods. There's American barrels. All bourbon's made with new American barrels. You don't reuse them. Wine, you can use a barrel several times. Barrels cost from $300 to $2,000 a piece. So a lot of the value of wine, if you get a really good wine, is it's been stored in a $1,500 barrel for 2 years, which costs a lot of money, and that adds to the, hopefully, to the flavor. And in the case of bourbon, these are open. If these are wine barrels, they'd be inside a temperature-controlled facility. Okay. Now, I want to finish up with this. This is everything we've talked about. So what happens? If you can see this, in this case, the gibberellic acid binds to a membrane-bound protein, which we've talked about, an integral protein. And when it binds to that protein, it causes the production of a product that activates some other proteins. And this is called a signaling molecule. So the GA never even gets inside the cell. And what it does is it causes, that GA causes a cascade of things to happen. So GA binds and releases an intermediate, which then goes into the nucleus. And I'm not going to go through the details here, but it binds to the nucleus in such a way that it produces alpha amylase. Okay? So we've gone from this activated intermediate coming into and binding to the DELA repressor to basically what we just talked about in bacteria, activating the genes and producing a gene product. And in this case, the gene product is alpha amylase. What's alpha amylase do? Come on, somebody tell me. Breaks down to starch. So how does it get to the starch? The alpha amylase RNA moves into the rough endoplasmic reticulum. What happens in the rough endoplasmic reticulum? What's the first thing that happens? What is RER? What's the function of RER? Oh, come on, folks. You just had that. What does rough endoplasmic reticulum, what mostly happens on the rough endoplasmic reticulum? Oh. Huh? Ribosomes. But what do ribosomes do? Protein synthesis. Remember? Good night. Wow. Good thing the exam wasn't today. So you have this alpha amylase RNA moving through, in this case, the nuclear membrane pores, producing the RNA, producing the protein, which then goes to the Golgi body, okay? And remember, I told you that the corn seed was just this mostly just bag of starch. It's not cellular, it's just a bunch of starch and other things, of course. Ooh, and I'm going overtime. And then the alpha amylase is bagged up by the Golgi body and then goes into the coalesces with the cell membrane, and the alpha amylase ends up here. Okay? We're over time. I'll go over this again on Wednesday.